Good evening, dear students. Hope all of you are doing safe and doing good. So today we will study about the other viral disease, that is rabies. Discussing about the disease, what the disease is about, which is the causative organism, and which organ does it affect in humans, the classification of that particular virus, the antigenic structure, viral and antigenic structure, cultural characteristics, pathogenicity, pathogenesis, symptoms, diagnosis, epidemiology, prophylaxis, and treatment. Rabies, as many of you would know, is an infectious viral disease of the central nervous system in humans. So, it is basically transmitted by dogs. So, dogs are the primary mode of transmission to humans. So, it is spread to people through bites or scratches, usually through saliva of the infected dog. So, although effective human vaccines and immunoglobulins exist for rabies, they are not readily available or accessible, especially for those in need. However, this disease can be prevented by vaccination to both dogs as well as to humans. So, looking at the classification of this organism, it belongs to the genus Lissavirus, family Rhabdoviridae, and the order Mononegavirales. Okay, this particular genus, that is Lissavirus, is delineated into different like different virus species based on certain criteria like genetic distance, immunological features, and antigenic properties, and also with the monoclonal antibodies that they can raise. And one more interesting thing is bats are the primary or sole reservoir hosts for all of the Lissa viruses except a very few. So when we look at the structure of this rabies virus, it is a rod or bullet shaped virus. It is it is having a single stranded RNA, that is negative sense single stranded RNA, which is non segmented, and it is also an enveloped virus. Okay, the virus measures approximately 60 to 180 nanometer in length or in the area. The viral structure is composed of an internal protein core or nucleocapsid. Okay, so this nucleocapsid contains the nucleic acid. It contains three components, basically the nucleic acid, an outer envelope which is surrounded by the lipid containing bilayer that is covered with a transmembrane glycoprotein spikes. Okay, so the genome of this rabies virus, the negative sense single stranded RNA, encodes five proteins associated which are associated with the ribonucleoprotein complex or the viral envelope. So if you see here, this is the envelope. So there are five proteins that can be encoded by this genome. Okay, so these five proteins, what are these five proteins, we'll see. So this ribonucleoprotein complex, as I mentioned, comprises of three proteins. That is, one is the L protein. This blue colored is the L protein, that is nothing but transcriptase. And this N protein, this gray colored what you see here, is the nucleoprotein or the nucleocapsid protein. And NS or the P protein, what you see here, is the transcriptase associated protein. Okay, so these three proteins, that is L, N and P, together comprise the RNP complex. Then these aggregate in the cytoplasm of the virus infected neurons. Okay, and this RNP complex together forms what is called as the Negri bodies. This Negri bodies can be stained and it can be used as a means of diagnosis. So we'll see the diagnosis part later. And coming back to the structure, so we saw with the RNP complex, the next two set of proteins are, one is M protein, that is the matrix protein, okay, and G protein, which is the glycoprotein, that is the spike protein. What you see here, the green colored one is the spike protein, and this dark orange colored is the RNA. So this is about the structure of. So looking at the cultural characteristics of this rabies virus, we need to know two terms basically, that is the street virus and fixed virus. So what exactly is this street virus? It is the freshly isolated rabies virus from natural infections, is referred to as either infections of either humans or animals, is referred to as the street virus. So after several serial intracerebral passages of this virus in the rabbit, undergoes certain alterations and becomes what is called as the fixed virus. Okay, so these are the two terms that we need to know. And in both these street virus and the fixed virus form, generally we can observe, or what can be observed is the Negri bodies that are seen in the brains of these laboratory animals of both the street virus and fixed virus infection, and these Negri bodies are used as a source of diagnosis in case of rabies infection. Rabies virus can be cultured in chick embryos. So as we know, this viruses require animal tissue for their growth, or they cannot be grown on artificial culture media. Hence chick embryos or any animal tissues are required, live animal tissues are required. So the virus is usually inoculated into the yolk sac of the embryo, and serial propagation in this chick embryo has led to the development of two attenuated vaccines that are referred to as Flurry and Caliph. And there are also several primary and continuous cell cultures that are used for the growth of rabies virus. So the fixed virus strains can be grown in human diploid lung fibroblasts, chick embryo and Vero monkey kidney cell cultures. At the pathogenicity, looking at the pathogenicity of this disease, once inoculated, so or once the virus enters the humans through animal bite, the virus replicates in the muscle at the site of bite, or it may travel to the peripheral nervous system directly. So if it like virus infects the nerves in the peripheral nervous system and then moves by retrograde transport, then the virus replicates in the dorsal root ganglion and then travels up the spinal cord to reach the brain. So finally the brain is affected, and the virus then again from the brain it travels through the nerves to other tissues and affects other tissues again, like eyes, kidneys and also the salivary glands. So this is the pathogenicity pattern of rabies virus. So we'll now look at the life cycle or the replication of this rabies virus. So we'll go stepwise and see what happens in each step. So there are 9 steps. So first one is the adsorption. So the replication proceeds with significant interaction of the G protein, that is the spike protein of the virus, with the acetylcholine receptors of the peripheral nervous system. Okay, so here the host cell is the nerve cell of the peripheral nervous system. So the G protein of the virus interacts or attaches to the acetylcholine receptors present on the nerve cell of the humans. The virus then gains entry to the central nervous system, that is the second step, that is penetration, and this happens by receptor mediated endocytosis. So because the virus is entering the cell through a receptor attachment. So within this endosome, the pH lowers, that brings about conformational changes in the spike protein and promotes the fusion of the viral membrane to the endosomal membrane. So because of this fusion, the viral nucleocapsid is released, and that is the third step, as what you see here is as uncoating. So the next step is transcription. So this single stranded RNA genome is then transcribed into positive sense mRNA by viral RNA polymerase. Okay, and this mRNA codes for 5 virion proteins, as we discussed earlier, that is RNP, M and G protein. Okay, then this negative sense RNA genome again acts as a template for complementary positive sense positive sense RNA. So this step, that is the 7th step, what you are seeing is what we are discussing now. On the other hand, what happens? There is also the synthesis of proteins that is happening through the process of translation in the 5th step. So the viral proteins are getting synthesized, and there is also the processing of these viral proteins happening, and simultaneously there is also the synthesis of this G protein. So this G protein synthesis is initially initiated on the free ribosomes. However, the completion of these G protein synthesis and glycosylation has to occur in the endoplasmic reticulum and also in the Golgi apparatus. So that's why you are seeing this 6th step, that is processing. So the initiation happens in the ribosome, on the ribosomes, whereas the termination happens in the Golgi complex or the endoplasmic reticulum. Then the next step is the assembly. The NPL complex, that is the RNP complex, encapsulates negative stranded genomic RNA to form the RNP core. Okay, so what happened? We will just brief it up. So what happened was, there was a negative sense RNA which was transcribed to positive sense RNA. So this positive sense RNA served as mRNA template for the translation of all the viral proteins, and this positive sense RNA also served as template for the synthesis of the viral negative sense RNAs, which was used for the assembly step. Okay, so in the last step, what happens? The mRNP complex then binds with the glycoprotein, and the completed virus along with all the viral proteins and the J proteins along with the RNA gets assembled in the 8th step, what you are seeing there. And then the viral matrix protein forms a layer on the inner side of the envelope, whereas the viral glycoprotein forms outer layer and also forms the spikes. So after this, the virus buds through the plasma membrane of the host cell. So this is about the replication of rabies virus. Next, we'll see the pathogenesis. So, what happens in pathogenesis? We'll be seeing how it affects the host cell, or what are the host responses that are seen as a result of the rabies infection. So, once inoculated, after inoculation of this infectious saliva to the animals by bite, the virus may persist there in the site of entry, or it replicates in muscle tissue before progressing to the peripheral nervous system. So, neurotropism is a main feature that is being associated with the viral replication, and there is also, so after peripheral nerve entry, the virus then moves centripetally within the axons to the central nervous system through transportation by retrograde axonal flow. So here the incubation period is dependent on the distance between the site of bite and the central nervous system. So apart from this, the intensity of infection also depends on the age of the host, the immune status of the host, and the viral strain involved. So what happens in the central nervous system? The multiplication of the virus occurs in the grey matter of the brain and then spreads to the endoneurium of the Schwann cells. Then the virus spread may be facilitated by movements across cell to cell junctions. So after a period of multiplication, then it disseminates into tissues and organs through efferent neurons. So again it is spreading to all the organs. So this is the pathogen. Looking at the symptoms, in case of rabies, the disease is an acute fulminant fatal encephalitis. So what happens is, after an incubation period in humans that is typically 1 to 3 months, and it can be even less, like 1 week as well. So the clinical symptoms here can be divided into 3 phases, that is a short prodromal phase, an acute neurologic phase, and coma. So we'll see phase wise what are the symptoms. In the prodromal phase, that lasts from 2 to 10 days, it may include symptoms like malaise, anorexia, headache, photophobia, nausea and vomiting, sore throat, and even fever. And during the neurologic phase, that lasts from 2 to 7 days, the patients may show signs of nervous system dysfunction like nervousness, apprehension, hallucinations, and etc. And apart from this, general sympathetic hyperactivity is being observed, including lacrimation and pupillary dilatation and increased salivation and perspiration. There can also be hydrophobia, that is fear of water, or aerophobia, that is fear when feeling a breeze, is exhibited by certain number of patients. And the act of swallowing precipitates a painful spasm in the throat muscles. Then the neurologic phase is followed by the convulsive seizures or coma, and it finally leads to death. The major cause of death is basically because of cardiorespiratory arrest. The disease course is slower, with sometimes the patients surviving for 30 days. However, recovery and survival are extremely rare. So, looking at the diagnosis, the specimen that have been used for the diagnosis are saliva, corneal biopsy, the brain tissue, neck skin biopsy. Excuse me. So these are the specimens that can be used for diagnosis. So when we look at the histopathology, that is when a tissue is stained. So as I mentioned earlier, diagnosis, one of the means the histopathology diagnosis, what is seen here is the, what is looked for is the nigri bodies. Okay, so these nigri bodies are stained by Seller's staining technique that comprises of basic fustian and methylene blue. So once when stained with these, with the Seller's staining method, the nigri bodies are purplish pink in color, that are sharply demarcated, and they have a distinct internal structure with basophilic granules in the eosinophilic matrix. So this photograph what you see here is the nigri bodies that have been stained. So the other antigenic or the other immunological methods and molecular methods that are available for the diagnosis of this rabies virus are as follows. So one is the antigen detection, directly detecting the tissues infected with the rabies virus are currently identified most rapidly and accurately by means of immunofluorescence or immunoperoxidase staining using anti-rabies monoclonal antibodies. The next one is the antibody detection, wherein the antibodies develop slowly in the infected persons or animals during progression of the disease. And serum antibodies to rabies can also be detected by immunofluorescence or neutralization tests. And another method is the isolation method. Available tissue is inoculated intracerebrally into suckling mice, and infection in mice results in encephalitis and also death. So the central nervous system of the inoculated animal is then examined for nigri bodies and also the rabies antigen. Then molecular methods like reverse transcription polymerase chain reaction testing can also be used to amplify parts of the rabies virus genome from fixed or unfixed brain tissue or saliva. So these are the diagnosis methods that have been available. Epidemiology of rabies virus. The infection is in man is generally acquired from the bite of an infected animal, specifically the domestic dogs, that is Canis familiaris, which acts as an important vector. Although there are bat rabies that continues to cause human infections in specific parts of the world like Central and South America. Then the virus still remains an endemic across much of the developing world, where the majority of the human deaths are due to rabies. Transmission, when we look at the transmission part, people are usually infected following a deep bite or scratch from an animal with rabies, and transmission to humans by rabid dog accounts for 99% of the cases. Transmission can also occur when infectious material such as saliva comes in direct contact with the human mucosa or fresh skin wounds. So that is where they gain entry, the virus gains entry. So what are the preventive measures? There are a set of preventive measures that are available. So the first thing what can be done is eliminating rabies in dogs itself, because they are the primary source of infection. So rabies is a vaccine preventable disease, and vaccinating dogs is the most cost effective strategy for preventing rabies. So dog vaccination reduces both deaths attributable to rabies and also the need for post exposure prophylaxis. So post exposure prophylaxis is the like what to say, the prevention of the disease once the humans are exposed to that particular virus. So that can also be avoided. And awareness on rabies and preventing dog bites, then also education on dog behavior and bite prevention, especially for children and adults, is an essential extension of rabies vaccination programs. And preventive immunization in people, that is the vaccines, human rabies vaccine exists for pre-exposure immunization. So these are recommended for people in certain high risk occupations like laboratory workers handling live rabies and rabies related viruses, and also for people whose professional or personal activities might bring them in direct contact with the bats, carnivores, or other mammals that may be infected with this rabies virus. Then, vaccination against rabies is used in two distinct situations: to protect those who are at risk of exposure and to prevent the development of the clinical symptoms, those who have already been exposed. So, the vaccination schedule involves a set of schedules that have been described. So, looking at the treatment, as in for rabies, there is no specific chemotherapy or any specific medicines that have been used for treatment. The only treatment that is available is post exposure prophylaxis, or preventing the disease after exposure of this virus, after exposure of this virus in the humans. So once the once any human is bit with this rabid dogs, then the first thing what has to be done is extensive washing. So washing of the wound for a minimum period of 15 minutes, either with soap or detergent or water or any iodine substances that can kill the rabies virus, and local treatment of the wound as soon as the exposure has happened, and a course of potent and effective rabies vaccine that meets World Health Organization standards. And also, in extreme cases, there can also be administration of rabies immunoglobulin, and a series of rabies vaccinations can also be help helpful to identify and fight these rabies virus. So stay away. The dog here, the rabbit dog here says, stay away from my bites, else vaccinate me to stay safe. And another message is, vaccinate yourself to stay safe from rabies infection. Thank you. If you have any doubts, you can post it in the course chat. Thank you.